Unveiling Identity, the step-by-step procedure of DNA fingerprint testing. Introduction. DNA fingerprinting, also known as DNA profiling or genetic fingerprinting, is a powerful tool used in forensic science, paternity testing, and genetic research. This essay provides an in-depth exploration of the step-by-step procedure involved in DNA fingerprint testing, highlighting its significance in solving crimes, establishing familial relationships, and advancing our understanding of genetics. Sample collection. The process begins with the collection of biological samples containing DNA, such as blood, saliva, hair follicles, semen, or tissue samples. Samples must be properly labeled, sealed, and preserved to prevent contamination and degradation. DNA extraction. In the laboratory, DNA is extracted from the collected samples using various techniques such as cell lysis, centrifugation, and purification. Enzymes and chemicals are used to break down cell membranes and proteins, releasing the DNA into solution. Polymerase chain reaction, PCR. PCR is a crucial step that amplifies specific regions of the DNA, making them easier to analyze. Primers designed to target specific DNA sequences are added to the extracted DNA along with DNA polymerase and nucleotides. The PCR machine cycles through a series of temperature changes, causing DNA strands to separate and new copies to be synthesized. Gel electrophoresis. The amplified DNA fragments are separated based on size using gel electrophoresis. The DNA samples are loaded into wells in a gel matrix and subjected to an electric field, causing them to migrate through the gel. Shorter fragments move faster and travel further through the gel, resulting in distinct bands that represent different DNA fragments. Staining and visualization. After electrophoresis, the DNA fragments in the gel are stained with fluorescent dyes or radioactive probes. The gel is then placed under UV light or X-ray film to visualize the DNA bands. The resulting DNA banding pattern, or DNA fingerprint, is unique to each individual, except identical twins, and can be used for identification purposes. Analysis and interpretation. The DNA fingerprint obtained from gel electrophoresis is analyzed to determine the number and size of DNA fragments at specific loci or regions. Scientists compare DNA fingerprints from different samples to identify similarities and differences. Statistical analysis is often employed to calculate the probability of two individuals having the same DNA profile. Database comparison. DNA profiles obtained from crime scene evidence are compared against DNA profiles stored in databases, such as CODIS, Combined DNA Index System. Matches between crime scene DNA and known individuals can provide valuable evidence in criminal investigations. Conclusion. DNA fingerprinting has revolutionized forensic science and has become an indispensable tool in criminal investigations, paternity testing, and genetic research. The step-by-step procedure outlined in this essay underscores the meticulous techniques and sophisticated technologies involved in DNA fingerprint testing. As advancements continue to enhance the accuracy and efficiency of DNA analysis, the potential for solving crimes and unraveling genetic mysteries grows exponentially, reaffirming the profound impact of DNA fingerprinting on society.